So this is the course that we are going to learn for this semester. As I correctly pointed out earlier, the continuation of the previous course, right? Sorry. Uh, can you all hear me?
Yes,
Yes,
sir.
sir.
Yeah.
Yes, sir.
Do you see my face as well?
Yes, sir.
Yes,
Okay.
Yes,
sir.
sir.
Yes, right. Okay. Simultaneously, you can see the PowerPoint presentation also, isn't it?
Yes, sir.
Right.
Yes, sir.
Since we are a little bit confused with the system, I was unable to send the PowerPoint well in advance. Sorry for that. Probably today or tomorrow, I will upload this in LMS, because actually speaking, I don't have a LMS account for Faculty of Agriculture, University of Peradeniya, so that I couldn't upload this to your LMS. But I will do it probably before the next lecture, right? Okay, so this is the course, Experimental Design. This is the 2-unit course, 30 hours. There's no any practical in it, but in class we will do some examples, practical examples. uh, Actually, if you look at this particular course is very, very, very important uh, for your final year research. So all of you would do a final year research after in another 6 months, uh, so that everybody will have a chance to go through at least some part of the experimental design that you are learning now. So this course is very important to all final year students. At least some of these aspects you will apply into your final year research. So that is why this course has been embedded to 4100 series, or in other term, 7th semester, right? Uh, just a second. Yeah, this course actually shared by myself and Dr. Sivachandran, Mrs. Sivachandran. As we did last year also, this course, half of the course will be taught by myself. The rest half will be taught by Dr. Sivamadi Sivachandran. So basically, this is I drawn from your prospectus. What are the objectives of this course? Students will furnish with knowledge and skill on designing of single factor and multiple factor experiments. We will talk about what is single factor, multiple factor later on. And also, once you do the experiment, you should be able to do the data collection as well as data analysis. Can you remember we defined the term statistics in your last course? What do you mean by statistics? Statistics basically comprise of 4 components. If somebody can remember, can you answer? 4 steps. It's a scientific matter of planning. It's a scientific matter of planning. Then, anyone? Everything gone with the corona, is it? Isn't it? I think so. Everything. Yeah, Indika can answer. Yes, Indika. Indika raised the hand. Yeah, you can answer. Unmute and answer. I expect the answer from Indika. Yes, Indika. I don't hear Indika. Anything wrong with my setup? Let's see. Can you all hear me first of all?
Yes, sir.
Yes,
Right.
sir.
Yes,
Okay.
Yes.
sir.
Yes.
Yes, sir.
Yes. Yes. What about Indika? Indika, can you answer? Can you all see my PowerPoint presentation?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes,
Yes, sir.
sir.
Yeah.
Yes,
Yes,
sir.
sir.
Yeah. So Indika raised the hand. Why? To answer. Yes, Indika.
Yes, sir.
Indika, yes, you raise your hand. Why? To give the answer, isn't it? Any problem? Oh, mistakenly you have raised. Okay. So, what do you mean by? Just a second. Right. What do you mean by the statistics? As I mentioned in previous course, statistics is actually planning your experiment. Then you collect the data. Then you classify the data as well as then you interpret the data and draw the conclusion. Right. So that is basically statistics. Here also we are going little further advanced than the very basic. We are going to do experiments with single factor as well as multiple factor, and how to collect the data, how to plan the experiment, how to collect the data, and how to interpret as well as how to come up with some conclusion of the data. So that is the concept of experimental design that we are going to learn here. Right. Learning outcomes. Learning outcomes. Uh, Actually, once we have learned this particular course, what are the things that has been expected from a student, right? We can translate the research objective into the hypothesis. Can you remember we learn about hypothesis? So we can formulate some hypothesis for your research. Uh, we will see with some example later on. Choose the appropriate design. When you say design, this is not the design as the dress design. Huh? For example, for dressmaking, we will have to have some kind of design. What do you mean by design for dressmaking? Anyone? Anyone could answer. What do you mean by design for dressmaking? Anyone? Anyone? What do you mean by design? Hmm? Anybody can? Yeah. So before you making the dress, you'll have to plan it. What is the size that you'll have to make the cut, right? So all these are called as designing. Okay. So here also, when you do or when you start an experiment, you'll have to choose a design, right? Choose a design. It is the roadmap to start the experiment and conduct the experiment. So with a design, a proper design or appropriate as well as proper design only, you will be able to execute any experiments. Then you analyze the data which has been taken from that experiment, and then you draw some meaningful conclusions from those data after the analysis of the data. Uh, for data analysis, I think you have another practical course. Probably I will also do some practical lectures, how to analyze the data, set of data, right? There are several softwares available nowadays. You don't need to do it manually. When we did our undergraduate, we did manually. That is in 90s, right? So that time the data softwares are not very popular, as well as we couldn't afford to purchase that high price data analysis tools. So that we did all by manually. But nowadays, you just feed the data into the computer in 20 seconds or 30 seconds, depend on the size of the data, you will get the figures or results. But you'll have to do the interpretation, and from that you'll have to make conclusion that computer doesn't do. That you'll have to make it by yourself. So that is the overall expectation from all the students from this particular course. Right. Uh, these are the course outlines. All these informations are available in your prospectus. Uh, Principle of experimental design, very basic. Then estimate of experimental error. This is the first design, that is called as complete randomized design. Then randomized complete block design, Latin square design, different types of designs are available. Then mean separation. Mean separation is done under some circumstance, not always. We will see later what are the circumstances need to be satisfied before we do the mean separation. Up to this point, I will be teaching. Beyond this point, Dr. Sivamati will be conducting the lectures. Interactions and factorial experiments. That is a vast area, very important and the vast area. And the modification of factorial experiments. Some of the modifications like split plot design, nested design, and so on. And also a topic on analysis of covariance. Covariance, very important when you do animal science experiments. The covariance are very important. How to quantify covariance. You can understand variance, right? Covariance mean it is the variance between two variables joined together, how they make a variation. Two variability between x and y, two variable, right? So that is basically called as covariations. So those are the lectures that course contain that we will addressing. Yeah, any questions? Right. One more thing I need to remind you regarding your what do you call um, what do you call this statistical table. I hope all of you have a statistical table, isn't it? All of you are having a statistical table. So today we don't need statistical table. From next class onwards, most probably we need the statistical table. So keep statistical table also along with that. Okay, right? Stay from next week onwards, we need to have a statistical table. Right. We can move to other section. that is on principle of experimental design. Principle of experimental design. Right? Just a second. Yeah. When you talk about principle of experimental design, mm, there are three principles that you should remember. First principle is randomization. Second principle is replication. Third principle is control of unexplained variability. Three principles that you should remember: randomization, replication, control of unexplained variability. Right? Okay. Randomization, replication, and control of unexplained variability. We will see one by one. Randomization. What do you mean by randomization? It is the procedure for allocating treatments into specified experimental unit. I will explain what is experimental unit little later. So allocation of treatment into specified experimental units, with each unit having an equal chance of being selected to receive a particular treatment. Just have a look. I will Okay, randomization. It is the procedure for allocating treatment into specified experimental units, with each unit having an equal chance of being selected to receive a particular treatment. So basically, what we are expecting here in randomization is the way that we are allocating treatment. In simple term, how are we going to allocate the treatment to the experimental unit? I will explain later what is experimental unit. So in what pattern or in what procedure that we are going to allocate treatment to the experimental unit, right? Uh, for example, uh, let's say I have an example here, animal experiment. I took an animal experiment. Uh, can somebody answer what do you mean by concentrate and roughage? Have you all learned this? Tanusha?
Concent-
Yes?
Yes, sir.
Yeah?
Concentrates of protein-based food items, and roughages contain much fiber, about
Okay,
more than
okay,
18%.
okay, right. Concentrate more contain with carbohydrate, basically. Roughage consists of more fiber, isn't it? Right?
Yes, sir.
Okay, right. I'm not the expert on animal science, but I just took as an example. Right, I'm going to do a small experiment. Suppose, let's say I'm going to do a small experiment, right? To evaluate the milk yield from cow. Uh, you see here in the first case, I'm going to take 70% concentrate in their feed and 30% roughage, which I am calling it as treatment 1, T1. Similarly, now I am taking another formulation, 60% concentrate and 40% roughage, which comprise of 100%, isn't it? Both together, which I'm naming it as treatment 2. Similarly, 50% concentrate, 50% roughage, which is treatment 3. 40% concentrate, 60% roughage, treatment 4. Okay? Do you understand? Yeah? So how many treatments are there? Now somebody can have to answer. How many treatments are there that I am going to test it?
4 treatments.
4
Four,
treatments.
sir.
Yes.
4 treatments.
Exactly. I have named it as T1, T2, T3, T4. Whereas all these have a unique combination. First one is having more concentrate, 70%, right? First treatment, second treatment, 60%, third treatment, 50%, 40%, and roughages other way around, lowest to upper value. So these are the treatments now that I'm going to test with the animals, right? Under what topic that we are going to learn this? Randomization. I'm going to look at randomization. What do you mean by randomization? Right. Now, how many treatments are there? As you all said, 4 treatments. So how many minimum cows need to try with this 4 treatment?
Four.
Yeah, I need at least 4, at least. Can I try with all this combination with one single cow?
Can't.
We can't, because all treatments are different, isn't it? So ultimate effect we cannot evaluate which is coming from which treatment. Therefore, we cannot use only single cow for all these treatments. So best thing is we'll have to use at least one cow per treatment. That understandable. Okay. Now, let's say getting these 4 cows. If I am going to take all these cows, 4 cows, if I select all these 4 cows from different breed, what do you think? If I take all 4 cows from different breed, my objective is to evaluate the milk yield, whether the milk yield is higher in treatment 1 or treatment 2 or treatment 3 or treatment 4. So that is my objective. Clear? So I want to evaluate whether this combination, first combination gives a higher milk yield, or second combination gives a higher milk yield, or third combination gives higher milk yield, and fourth combination gives higher milk yield. So that is my objective. Clear? So then I need to have 4 treatment. If I choose 4 different breeds, what is the problem? Let's say this is Frisian, this is Aisha, this is Saiwal, this is Sindhi. So what is the problem if I allocate all these 4 treatments to these 4 cows, which are very different from breeds? Normally, breed itself, do you think this Frisian gives the same milk yield as Aisha?
No,
Of a-
no,
No,
sir.
no.
No. Breed itself has some effect on milk yield, genetical difference, isn't it? Sahiwal has a different milk yield than the Frisian, for example. So already some genetical difference is there. if you apply this treatment to already genetically different material, the ultimate effect that we are going to evaluate, that is not only due to this uh, uh, feed experiment, but also due to the breed effects, isn't it? So in that circumstance, what is the best idea? If I
Same
want
breed.
Same breed.
exactly,
Same breeds.
exactly, you have 4 treatment, take all our same breeds, right? That is one thing. Now let's say, sorry, I have pressed that. Now let's say you have come to a conclusion, you are going to take all 4 cows from the same breed. That is fine. That is first step. Second step, milk yield, forget about this concentrate or roughage that you are going to give. uh, Practically as well as theoretically, milk yield based on the parity as well, isn't it?
Yes, sir.
Yeah, that, for example, milk yield, let's say this cow is with third parity, this is with second parity, this is with fifth parity, as an example. Can you expect all to give same amount of milk?
No, sir.
No,
No.
sir.
So the parity is, if the parity is already different, uh, or already that variability is there. So when you select 4 cows, we selected 4 cows from the same breed, same parity, right? Then most probably same weight. Usually very difficult to same age. Same age is possible, at least in terms of years. The fourth thing that you'll have to consider is more or less same weight, right? More or less same weight. Because why? Suppose let's say this cow is having 100 kilos. This cow is having 200 kilos, even though they are having same breed, same age, same parity. What is the problem? This cow consume more or less more feed than this cow, isn't it? This is, sorry, this is 100 kilos, this is 200 kilos. So 200 kilo cow definitely will consume more feed. If you have more feed consumption, automatically more milk, theoretically, isn't it? So because of that, all characters that we are selecting, or that we are considering in selecting cows, has to be more or less uniform. That keep in mind, right? But practically, I said 4 things. First is breed. We can get all 4 cows from the same breed, so no problem. Second, they are from same parity. That is also possible. We can get all 4 cows from the same parity. That is also possible. Third, age. Can we get all cows from the same age?
Yes,
Yeah,
sir.
that is also possible. Right. Fourth one, what I said is weight. Can we say all cows can be taken from same weight?
Not exactly.
Not exactly, because in your class, how many students are there? How many colleagues? 74 colleagues. 74 students are there. If I want to select 10 or 4 of them with 50 kilo exactly, 50 kilogram exactly in weight, can you get? Very difficult. So very, very, very difficult to have the animals with the same weight, right? You can get same breed, same age, no problem. Same parity. In a big farm, you can. But with the same age, sorry, same weight, no way at all. Right? Now, right? Let's say, sorry, let's say we have 4 cows: cow 1, cow 2, cow 3, cow 4, right? As I said, the fourth point where the weight is slightly differ from each cow. Others are same: parity, age, as well as the breed. All three are same, but except the weight. Now I am numbering these cows as 1, 2, and 3. Okay. 1, 2, and 3. 4. 4 cows. 1, 2, 3, and 4. Right? Now, as I pointed out, how I am going to allocate this 4 treatment among these 4 cows. Right? Suppose, let's say this is one. This is I cannot write on this. That is the problem. Sorry. Yeah, I cannot write. Okay, so we have 4 different cows. They are different slightly in their weight. Fine. They are slightly different in weight. Let's say cow 1 is having lowest weight. Cow 2 is having slightly higher weight. Cow 3 is having higher weight than cow 2. And cow 4 is having higher weight than cow 3. So in this, this is in ascending order. Cow 1 has the lowest weight. Cow 4 has the highest weight. There's a slight change in the weight, assume. Right. Now, how I am going to allocate this treatment among these 4 cows. For example, let's say if I am saying T1 for cow 1, T2 for cow 2, T3 for cow 3, and T4 for cow 4. Then there's a biasness, right? Why there's a biasness? Anyone? Because by looking at the cow, by looking at their weight, we are giving the treatment. So that is no any randomization. So what is the best way to randomize? For example, right, there's a small, yeah, right. What I do is I simply write down this as cow 1, this as cow 2, this as cow 3, this as cow 4, and putting into the small box cow 1, 2, 3, 4, 5, 4 small piece of papers in 4 different small piece of papers, right? Then I take the first paper. I say it is cow 4, right? Cow 4. So that means I am giving the first treatment to cow 4, because I'm not by looking at visually allocate the treatment. I am allocating the treatment randomly, right? Similarly, second time I take the piece of paper which says cow 1. So I am applying treatment to cow 1. So likewise, we can allocate all the treatments. So this is what we call as randomization. We didn't give any priority to any of those cows. Okay. So that is the example for randomization, right? Randomization without giving any biasness. We give equal chance. You see, we give equal chance to every cow, because that is why we have named all 4 cows and putting into the box. Then we have taken it one by one, giving equal chance. right? Actually, what is the purpose of doing this randomization? What is the purpose? The purpose of doing this randomization is to get the unbiased estimate. Get the unbiased estimate. So basically, what is the purpose or what is the objective of this randomization is to have unbiased estimate. What do you mean by estimate? Can you tell me what are the estimates? What are the estimates? Parameters. We learned about parameters. Can you remember? Population mean. How do we write population mean? How do we write population mean? Symbolically? Symbol.
Okay.
Yeah. How do you call population mean? Population mean, how do you write? I am unable to write on a pad here. That is why. Yeah. How do you write population mean? Mu, isn't it? Mu.
Yes, sir.
Yeah, mu. Population mean, we write it as mu. Those are parameters. Population mean is a parameter. Population variance. How do we call population variance in shortened form? Sigma
Scale.
square. Exactly. Sigma square. We have population variance sigma square. So these are parameters, population parameters. We call it as population mean as well as population variance are called as parameters. Then what do you mean by estimates? So estimates are coming from sample. For example, sample mean. Sample mean. What is the notation for sample mean?
X bar.
X bar, exactly. Right. So we learned that. So X bar is the sample mean. So sample mean is the estimate. So what is the objective of this randomization? What is the objective of randomization? We want to have unbiased estimates. What do you mean by unbiased estimates? Biasness, you know what is biasness? Giving priority or giving an opportunity to a particular aspects, right? Without giving equal chance to others. So that is called biasness. So unbiased estimate mean when you estimate, for example, could be population, sorry, sample mean or sample variance in other term, all these are called as estimates. So when you calculate, usually we do sampling and we do calculations. So in this value that you are calculating, suppose it is sample mean or sample variance. So these values will have high weightage. Why? Because it is coming from unbiased. For example, if I want to uh, talk about average weight of your class, right? Where I'm going to take 10 sample from that class with 74 students. And from that 10 sample, I'm going to come up with estimate, mean, X bar. Let's say, since I didn't have contact with all 74, I just take students from only living in Kilnochi or Jaffna, taking 10 students. If I take average of those 10 students of weight and say this is the average of whole class, is that correct? That is wrong, isn't it? Because it is biased. If I want to say that is the average weight of students living in Jaffna, that is fine. But my objective is I'm going to estimate the average or mean for the whole class, so that taking Jaffna group will not represent the whole class, right? Okay, right. We are having a small break, then we will continue. Right, in 5 minutes we will continue. Okay. Yes. Are you all back? Shall we continue? I
Yes,
couldn't.
sir.
Yeah.
Yes,
Shall
sir.
so you had a you all are at home. I'm at office. You see, I'm at Kilnochi. Yeah. So right, fine. So basically, randomization is how or what is the pattern that we are going to allocate the treatment to experimental unit. Right. I said I want to explain this experimental unit. Right. Right. Look at the previous example. Uh, experimental unit is actually it is the unit that uniquely treatment applied. Experimental unit is the uniquely treatment applied. Now here, how many cows are there? 4 cows.
Focus.
So each cow will all are they all 4 cows receiving the same treatment or different treatment?
Different.
Different. So for example, cow 1 receive treatment 2, cow 2 will receive treatment 3, cow 3 will receive treatment 4, and cow 4 will receive treatment 1. So here what we can say about this cow 1 is called as one experimental unit because it is receiving a unique treatment, isn't it? So that cow 1 is an experimental unit, cow 2 is another experimental unit, cow 3 is another experimental unit, cow 4 is another experimental unit. So each of these cow basically called as experimental unit. Okay. Unfortunately, I cannot draw here. In agronomy experiments, when you have plots in the field, right? When you have plots in the field, I'm not sure whether I can. Yeah, that's something else. Is when you have plots in the field, okay? This, just a second. I'm also trying with new technology. That is the problem. Yeah. Okay. When you have plots in the field, each plot, for example, 1 into 1 meter square meter, for example, that is named as one single plot. 1 into 1 square meter. In 1 into 1 square meter, how many tomato plants that you can plant according to the spacing? Agronomy specialist, specializing students in agronomy. Can you tell me, let's say 2 into 2 meter square plot, 2 into 2 meter square plot, that means 4 square meter. In 4 square meter, how many tomato plants that you can plant it? Yeah. Anyone? Example, what is the spacing of tomato plants? 5 into 5 centimeter. What is the spacing of tomato plants? Anyone? Agronomy group. No one is there. Yeah, let's say if the spacing is, I'm also not expert on that spacing. Let's say 30 centimeter into 40 centimeter, right? So 30 into 40 centimeter, that is, so based on that, you can calculate how many plants that we can accommodate within a plot, 2 into 2 meter plot. I'm unable to draw it. From next week onward, we can draw using the board. Right. So in that 4 square meter, we can calculate how many plants that we can accommodate. Right. A single plot, within a single plot, we can calculate how many plants that we can accommodate. So all the plants within that plot will receive a single treatment. Let's say we have 4 fertilizer, different composition of fertilizer, similar to this, 4 different composition of fertilizer. So we need 4 plots like that. 2 into 2 meter plot 1, 2 into 2 meter plot, second one, third one, and fourth one. We need 4 plots. So each plot will receive a single treatment or single treatment combination of fertilizer. So that is how we call it as experimental unit in if you are doing any field experiment, that is called as experimental unit. Normally, not like in animals. In animals, one cow can be a single experimental unit, no problem. Maybe in animal science, one hen or chick can be experimental unit. But usually in doing poultry, we will have more than one. Why? Sometimes if one bird dies, then chicks especially, then we have problem losing the information, isn't it? So that we will have more. Maybe one tree, a grown tree, can be considered as one experimental unit. But in field crops, usually we don't consider one field crop, one single field crop as experimental unit. Collectively, several number of field crops. That mean, several number of plants in a single field crop is called as plot. As I pointed out earlier, 2 into 2 meter plot, we can have roughly about, let's say, 20 plants, roughly about 20 plants, let's say. So all these 20 plants within the plot will receive the same treatment. So that is called as experimental unit, right? So experimental unit here is that, all right? When you do this randomization, by doing randomization, as we already said, we can go for unbiased estimate. That is the major objective of this randomization. And meantime, parallelly, we can ensure there is an independence among the observation. Observations are independent, basically. So then only we can do the randomization, right? So to have observations are independent, they should be more or less homogeneous. They should be more or less homogeneous. Yeah. Some people are switching on the microphone. Please switch off the microphones. Huh? Otherwise, we are getting disturbances. Whenever you want to answer, you can switch on. Independence among the observations, and at last, required for valid significant test and interval estimates. So later we will see how we can do the significant test. Already we have done in our previous course t-test, one sample t-test, two sample dependent and independent t-test. So all these tests will be based on the valid estimates. So if you have a valid estimate, then we can have a proper, a good, valid, significant test as well. And we can formulate intervals also. Confident interval, can you remember? Yes, we can formulate confident interval also. Right. This example we have seen. Now we will come to other topic. What do you mean by replication? This is the second point of principle of experimental design. First one is randomization. Second one is replication. What do you mean by replication? Replication. What do you mean by replication? So earlier in this example, Can you tell me how many times treatment 1 appears?
One
Ye-
time.
Only
One
once,
Time.
one time. So treatment 1 is appearing only one time. Treatment 2 is appearing only one time. Treatment 3 is appearing only one time. 4 is also appearing one time. So this is only one replicate. This is called as one replicate, right? All the treatments are appearing only once, so that it is one replicate. So, but with one replicate, we cannot do the experiment. With one replicate, we cannot do the experiment. Therefore, we need to repeat the same experiment again in a similar way. Is it clear? So we will need to repeat the same experiment with another set. So that is called as replication. You repeat it. For example, this is the plot I said. This is plot number 1. This is plot number 2, plot number 3, plot number 4. Right? This is one set of plot. This is another set of plot. This is the third set of plot. Basically, these are called as blocks. We will come to that later. What do you mean by blocks? So this is first set of plots. In the second set of plot, third set of plot. So every time, let's say this is the first treatment A appearing in all 3 sets of plots. So how many times A appearing?
3 times.
Yeah. So that means what is the replicate number? 3. Replicate is 3. 3 times it is repeated. The same treatment repeated 3 times. Look at here again. Same story. Treatment B repeated again 3 times. Treatment C repeated 3 times. And treatment D repeated 3 times. So all are repeated. How many treatments are there? How many treatments? How many
four,
treatments?
four
treatments.
treatments.
Yeah,
4 treatments.
exactly. 4 treatments. Yeah, sorry. 4 treatment. 4 treatment. Each of them named as A, B, C, and D. A is appearing 3 times. B is appearing 3 times. D is appearing 3 times. C is appearing 3 times. So what is the replicate now? What is the size of the replicate? How many times they have been repeated?
3 times.
3 times. So here we call it as 3 replicate. So any experiments will have to have minimum 2 replicate. Minimum, I said. The decision on replication depends on so many factors, but minimum will have to have 2 replicate for any experiments. When you do, could be water sample or could be soil sample. I said minimum, but maximum could be any number that will tells you a little later how we decide the number of replicates. Right. It is again the visual explanation, the same explanation as we did. This is how many replicates? This is the replicate number 1. Why? Because all the 4 treatments are appearing there. Treatment 1, 2, 3, 4. Similarly, we have another set with all 4 treatments. So this is replicate 1. This is replicate 2. Likewise, we can have several replicates. So we said we should have minimum replicate as 2. Right? Okay. Right. Replication again further expanded. Each treatment is applied independently to 2 or more experimental units. That is what we said. Each treatment is applied independently to 2 or more experimental units. So minimum is 2, as I said. Variation among plots treated alike can be measured error variance. This is more. This is one of the important points. You can note it down. Right? Why we want to do the replication? Why we want to do the replication? Can somebody explain to me why we want to do the replication? Can't we do experiments with a single replication? Why we need more than two, or two or more than two replication?
To reduce number of errors.
Yeah, yeah. One way, one thing is to reduce the error. Error mean actually error variance, right? So suppose if you have sample, if you have 5 sample versus 10 sample from a population, right? 5 versus 10 sample size. In one population, you have 5 sample, another from the same population, but this time you have 10 sample. So which one is better actually? 5 sample one from a population, or 10 sample from a population to have a good best estimate?
10.
10 sample.
10. Yeah. So when sample size increases, variance decreases. So the value estimate is coming much closer to the population value, right? That we learned in the previous course. So when you increase the sample size, that is fine. Right. My question here is, what we measure among this between two replicate? I said that there is one replicate, there's another replicate which is treated alike. For example, in this case, I'm going to measure the variance because this cow is treated with treatment 1. This cow is treated with treatment 1. So I'm going to calculate, suppose I have another set here, third set, third replicate, where one cow treated with treatment 1. So this cow plus this cow plus this cow. If I calculate the variance, what does it mean actually? What does it mean? Because here also I am applying treatment 1. Here also I am applying treatment 1. In one of the cow here is also I am applying treatment 1. If I measure the variance between these 3 cows, right, you will have some value of variance. What does it mean? Because I'm treating with the same treatment. I'm giving the same amount of feed, same percentage of this roughage and concentrate, but my milk yield coming differently. Why? That we cannot explain. That is called as error variance, right? In the last class also I mentioned about how what is this error variance. When you take tomato plants, different tomato plants, if you give all conditions similar except you treat them a different fertilizer mixtures. So right, suppose if you give with the same fertilizer mixture 4 different plants, all these 4 plants are having same genetical material, the ultimate yield will be differs. So that is why we call it as that term as error. The error is actually measured variance among the plot treated alike can be measured, which is called as error variance, right? As I pointed out earlier, so this cow is treated with same treatment T1. This cow is again treated with T1. Let's say another third set cow that is also treated with T1. If you measure the variance between these 3 T1 treated cows, the variance that you measure is called as error variance, which cannot be explained basically, right? By human beings, we cannot explain variance among the plots treated alike can be measured error variance, which if you have, if you increase the replication, it increase the precision. Somebody already said I took an example, 5 sample versus 10 sample. This is the case. Increase the precision, right? 10 sample is better than taking 10 sample is better than 5 sample, right? So as n increases, in other term, the error variance decreases. I mean, basically variance decreases. So error variance decreases when you increase the sample size. Okay. So because of that, replications are important. So here the main point why we are doing replication is to obtain the, you can write down why we are doing replication, to obtain the error variance. To obtain the error variance, we do replication. What is the usefulness of this error variance? We will talk about little later, maybe today or next time, right? So why we want to have error variance? Some of you might think about it. If it is say it is error, so why should we have error then in our experiments? We need to have some error, right? But not purposefully. This is by automatically coming when you treat the same treatment for different experimental unit, you get the error variance, right? Okay, so right, probably this may be the last slide that we will put forward. All right, what determines the number of replications? What determine the number of replications? Because I said the minimum is 2, maximum any number. But we cannot simply go for maximum. No. I said in cow experiments, minimum is 2 sets. Can you go for 20 sets? Very difficult. Finding cows will be a difficult, isn't it? If you can have, no problem, but finding sets are difficult. Right. Pattern and magnitude of variability in the study area. So pattern and magnitude of variability in the study area. We will talk about this in second type of design, pattern and the magnitude of variability in the study area. For example, even in this cows, right? If you look at the pattern of the variability, let's say this breed is same, age is same, parity is same, but what is the difference that I said? Weight. We cannot find same weight cows, but let's say this cow is 190 kilo, this is 198, this is 200 kilo, this is 205 kilos. So this is an increasing gradient in this direction. So there's a small gradient, isn't it? There's a small gradient. This is what we said here, magnitude of the variability, pattern and the magnitude of the variability. So we'll have to study about the pattern and the magnitude. So here the pattern, weight pattern is increasing, and increasing by how fold? For example, this cow is 150 kilos. This cow is 175 kilo. This is 202 kilos. This is 228 kilos. Look at this variability in this set. What do you think? Variability in this set versus variability in this set. Which set is higher in variability? Here I gave some values, right? Here also I gave some values. So here the variability is very high. Why? The big numbers. Here 150, here 175, this 198, is 228, something like that. So the variability is high in this set. So we'll have to decide how this variability and the magnitude of the variability gives you an idea to select the number of replications. This is true in among the animals, but if you go for some agronomy experiments, even in the field, there are some problems, right? For example, uh, the fertility level of the soil at a particular place. Let's say at this particular place, you measure the fertility or cation exchange capacity, amount of nitrogen, phosphorus, potash, all you can measure. You walk 10 meters away from this point. Can you tell me exactly whether you will have the same value as this point of all what you have measured here? Definitely there will be a different level amount of phosphorus, different level of nitrogen, different level of potash, different level of cation exchange capacity. So place to place, it varies. Even in the soil, soil has a huge variation. Maybe people who are specializing in soil science would talk about much on this spatial variability of soil, right? So, so we'll have to study all these variability, and based on that, we can decide the replications, right? So, I mean, that is also one of the important criteria to decide how many replications that we need. Okay, right. So, that is one thing. We will talk about this further when we discuss different types of design as well. The other one is number of treatment. The number of replicates is usually depends on number of treatment also. If you have less number of treatment, we can go for slightly larger number of replicates. If you have large number of treatments, then very difficult to go for large number of replicates as well. Then size of the difference to be detected. So that is also important. The depend on the size of the difference only we can say whether it is significant or not. So size of the difference that we need to be detected, it's depend. If the size of the difference to be detected is larger, right? Then we can go. I mean, it depends on the value. For example, you are taking uh, water sample from a pond or flowing river. You are taking water sample from flowing river at a particular time, right? Maybe two sample is fair enough from a flowing river when there is no rain. Say you take two sample is fine, right? So that is how we decide. Suppose during the rainy season, during in the rainy season, if you take water sample, you need to have more water sample. Why? Somebody can explain? Because depend on the way that it is finding the riffle and so on. The water is mixed with sand, other debris material. If there's a huge rainfall, so I mean, you need more sample because in one set of sample, the composition might vary. So you need more sample during that period. If the water flows without any disturbance, by runoff or whatever it is, then small sample is fine. Depend on required significant level. So that is related to the difference that you want to mark it. Amount of resources that can be devoted to the experiment. That is also coming in. How much resources that we want to devote it. If we can, resources mean, for example, finding cows, same breed, same parity, same age. Can you find because for treatment, if I want to have 5 replicate, how many experimental unit or number of cows needed for treatment? 5 replicate, how many cows with the same condition needed?
20.
20. So can you find out from a particular farm 20 cows with everything is same? Difficult. So depend on the resources, right? So that is what we say. Maybe maximum we can provide 12 cows, 3 replicate, right? And also ultimately, as usual to other cases as well, cost, limitation in cost. So definitely there are cost involved in any kind of experiment. So how we can provide that much of cost as well as labor, time, and so on. So the number of replicate depends on that as well. If you need more accuracy, we need to pump more cost, or more money, more labor, more time, and so on. So these are some of the drawbacks as well. Right. The third important factor in the experimental design, or the principle of design, is called as control of experimental error. Control of experimental error. We will continue this lecture from next week onwards. Next time we are meeting Tuesday 1 to 3. Tuesday 1 to 3. Okay, next time Thursday we are not going to meet. Instead of that, we are going to meet on Tuesday 1 to 3. Hope we will have more interactive board for writing and so on. So then we can have a better interactive session. Right. Any questions? Maybe we spend about 5 minutes to ask any questions. Anyone would like to ask questions? Yeah. Anyone would like to ask questions? Can ask. Yeah. Then we will. No questions, huh? Everything is perfectly understood, huh? So shall we have a quiz then? Shall we have a quiz? There is a program. Hello. There's a program called Perisol. I don't know how many of you have used it. Even I haven't used it. In Perisol, you can have a quiz. You can work from home. Every moment of your answering will be appearing on the screen here. So by copying or whatever it is, everything will appear. Shall we have a quiz then? Huh? Anyone? People are keeping some. Huh? Hello? No, all are silent. Why? Hello? Right. I think we will stop with that. We will meet next week. Okay? Right.
Thank you.